introduce Liz. I think many of you already know her, but she did her PhD in chemistry at Emory University. We do postdoc work at USC and at Caltech and Grant Johnson's lab. Then she started a lab at Emory and serves as the director of the EM core there. And now she has started at the University of Wisconsin-Madison. She started there last year as a full professor and director of their EM facility. So it's also a special pleasure for me to introduce her because I did my PhD in her lab. So yes, thanks Liz for coming, and we're excited to hear about virus assembly.
Well, thank you, Rebecca. It's a real treat to be here and see how well you're doing, and thank you for letting us spend some time with the NeCEN team today and yesterday. So I'll talk a little bit about the stuff that we've done on studying measles virus assembly and looking at the virus in general. This is completely different than what I've talked about previously here, so hopefully you'll enjoy it. One of the major things that many of us are doing in the field, and something that we focus a lot of attention on in the group, is thinking about how we prepare samples. And so for most of the work we have done, we have focused on purifying viruses and then imaging them at various states. But this really puts them out of context, and so it can be kind of problematic. And so what we've been trying to develop in the group is a technology that allows us to seamlessly go from a virus-infected sample all the way through every imaging methodology that we would like to do, so light-level imaging to EM-level imaging. This doesn't come without hardships, of course, because we may not be able to do every physiological experiment on them, but ideally what I would like to tell people is if I'm showing them a virus either in this state by light-level imaging or by EM, that the virus they're looking at is actually an infectious virus particle versus just saying, there's a population of infectious virus particles on this grid, but I can't tell you which one that is. So that is our goal for our studies of various viruses that we look at. So I'll highlight a few things we've been doing. It's hard to believe it's about 10 years that we've been working on measles virus. Our initial studies that we did, looking at the virus by cryo-EM and tomography, moving into the whole cell cryo-ET, and a little bit about what we're thinking about for the future. Forgive me, I seem to have a little cold, so if I'm not talking super loud, I apologize. So we know that measles is a extremely contagious virus. It's an airborne pathogen. We've had a lot of trouble lately with individuals through maybe religious preferences or personal preferences not vaccinating their children. So there's been there's been a higher proportion of measles cases worldwide, leading to deaths or just severe infection. And so even just last night, I was pulling numbers and thinking, oh my goodness, we're deploying the Red Cross to take care of a lot of outbreaks across Europe. And in the U.S., we're barely into 2019, and we've got a significant number of cases. And so the challenge with measles virus is, of course, there's a vaccine against the virus, but people are choosing not to vaccinate their children. And so what happens is we don't have herd immunity helping us regulate, combat the virus. And so this is really critical because herd immunity for measles virus, you need to have about 95% of your population vaccinated for that to take effect. And this is important for people who say are immunocompromised and maybe can't be vaccinated. And so if we have a population that's changing because we're moving a lot and it's dynamic, we can't inhibit the production of the virus and the infection process. So if you are a healthy individual, I encourage vaccination. I know in the lab at Emory we had to be vaccinated because of working in clinical space, but I still continue to encourage that with various group members. So we've been studying a lot of paramyxo and pneumoviruses in the group, and these are major pathogens to human, especially the geriatric and pediatric populations. And so since we were in a department of pediatrics, we kind of focused on infectious diseases related to children. We have a number of representative viruses, measles virus, which I'll talk about here, parainfluenza, human metapneumovirus, and RSV. So we focused some attention on RSV as well, but I won't talk about that today. So these are negative sense non-segmented single-stranded RNA genome viruses, where if we're looking at the genome of our measles virus, we have the nucleocapsid protein, the phosphoprotein matrix, F, which is the fusion glycoprotein, H, which is the attachment glycoprotein, and L, which is the polymerase. So if we look at this kind of cartoon about how these viruses should actually look and assemble, you'll have the two surface glycoproteins. The virus is surrounded by a lipid envelope derived from the host cell, the matrix protein, and somewhere, amazingly within there, there's the ribonuclear protein complex. So N, P, and come together, and L, and then you have N and the RNA genome interacting, and the genome's encapsulated into N. And so in the group, we've really focused a lot of attention on studying the processes on the two ends of the virus replication cycle, so namely entry and the presence of the glycoproteins on the surface, and then the assembly process, so really what's how matrix is governing the virus's assembly, and then the released virus, its structure at the back end. The reason we haven't focused on very much attention on this is you have one of those tools downstairs where we haven't had access to a milling system to allow us to thin the samples to be able to capture those events within the host cell. Now, we will have those tools, and so that's the next foray into measles virus and RSV replication cycle. So as I mentioned, our objectives for this project, and these are long-standing, 10-year-long objectives, to really determine the native state arrangement and structure of the glycoproteins on the surface of these viruses. So we know they're ordered on the surface of the virus particles, but the relationship space within them, so how many Fs to H or H glycoproteins are required to regulate the attachment and fusion process with the host cell receptors and the membrane. And then, of course, to understand how the matrix protein was regulating virus structure and the coordination of the other macromolecular complexes or structural proteins in the virus. Was it in fact lining the membrane, as many of these pictures have outlined, or did it have some other conformational state that we just didn't understand? So these were really target structural questions we had in the lab, not only looking at measles virus, but the other paramyxoid viruses that we were studying. And so this was really early work, in 2009 when we were going away to collect data, and we'll go away and collect data again now until we get our microscopes at Madison. And so this is what a frozen grid of measles virus looks like. And so this is a very high titer purified virus, so this is like 10 to the 7th virus, and a small aliquot of a sample, and it had been sucrose cushion or gradient purified, so it was really supposed to be ultra pure. But if I was to ask any of you which one of these particles on this grid was a virus particle, and that you were actually going to want to collect data there, we could not identify them, because these viruses are cell-associated viruses, so a lot of cellular debris also is present no matter what we would do with purification in the preparation. Now, luckily, we did we did figure out how to actually search well on these grids and find viruses, and so that was one of the viruses that we were happy to see in the beginning. And so if you remember that cartoon that I showed, I'll go to a central slice if this guy doesn't go crazy. Controls are like micro size. So if we look at this particle, you can see that there's these great glycoproteins on the surface. There does appear to be some thickening of the membrane here, maybe highlighting that there's matrix protein there, and then the ribonuclear protein complex that's circulating throughout the virus particle. But this wasn't ideal because of the challenges faced with collecting this data. And so our goal was to move not only technologically with the microscope into a space where we could collect really much higher quality data, but also to improve how we were preparing samples to support our ability to understand the structure of these viruses. And so in many cases, everyone in this room probably has gone through processes of purifying a virus, a protein complex, or even centrifuging down bacteria so you can concentrate them for applying them to an EM grid. And so we all know that these can cause damage, like to the villi on the surface of these guys, or flagella, or you can alter the structure of your protein. And one of the things we were very concerned about, because, as I mentioned, measles virus is an extremely pleomorphic enveloped virus. And so one of our concerns was, through any of these processes, which were pretty harsh, of purifying these particles, were we causing some sort of damage to the actual structure of the virus that we so desperately wanted to understand? And, you know, in addition, I mentioned that these guys grow to very low titers, so 10 to the 7th is a good day. You know, if you're thinking of a general icosahedral virus, 10 to the 11th or 12th is you know, your target. So we were really pushing it. So we wanted to figure out if there were other ways we could purify these viruses or capture them on the grids so that we could collect data. And so just to highlight a rogues gallery of other viruses we studied, you can see here are some of the other challenges we face with looking at these envelope viruses, especially in cryo-EM grids. So, you know, uneven distribution, you have particles, you know, arranging themselves around the quantifoil holes, which is really irritating because you want to maybe be able to average things, or if they're clumped together, maybe you're not going to be able to get good volumes, so you can do sub-volume averaging. Or in the case of Newcastle disease virus, like measles virus, it's also a very low titer virus particle that's being produced. So how can we get around some of these really super challenging things? Well, we can work on other systems, which we do work on in the lab, but we couldn't just totally give up on measles or RSV. We could think about how we could improve our methods of purifying them, stop relying on purification, or can we think of a way of capturing these particles on a grid so that we can minimize some of the earlier steps of handling? And so some of you may be aware of the originator of some of these technologies with Debbie Kelly when she was in Tom Waltz's lab, of developing this affinity purification or affinity grid technique. And so simply what this is you, and I'm not showing every single reference here, because there are now many, many papers that are showing different substrates you can use to adhere selectively an object of interest on a grid. So here, what it is you have a lipid layer, lipid monolayer that you apply to a grid, and you have your simple lipids in that monolayer, but then you also have the active nickel NTA lipids. And so by having that nickel NTA on there, you can think of it as an affinity column on your grid substrate, which is fantastic. So if you're wanting to purify a protein that has a his tag, or if you have an antibody to your target protein, either on its own or on the virus, in this case we were using the glycoproteins on the surface, we could use that to capture our object on the grid in a very selective manner. Obviously I'm showing you this cartoon of the perfect world in game, and we're not there yet, but we're very close in many labs of getting there. We can have this very regular arrangement of particles on the grid, and this will help eliminate some other problems that we're dealing with, like the air-liquid interface. So a number of people are improving this. So you've seen this picture on the left before, where it's an untreated grid, and so then we can look at a tomogram, slice through a tomogram of that, so we have all these cellular vesicles here, and this really represents what a field of viruses could look like, and the challenge we faced with having all these the cellular debris in that particular field, especially if you wanted to do sub-volume averaging on the glycoproteins on the surface, for example. But if we used the 20% nickel NTA, and so we used a number of different percentages of the nickel NTA, 50% was a little bit too much, 20 seemed to give us a good distribution, where we eliminate the trash in the background. You can see we have much fewer particles on the grid, but if we were counting those particles, we had a much higher proportion, or equivalent proportion, of the viruses present. So that would allow us to really more rapidly collect data. So you can see, again, the glycoproteins on the surface, there's hints of the RMP in here. So this was a much better place for us to be. And ideally, what you could do is even just you could dispense with purification through a sucrose cushion or gradient, and you could just flow lysate across this and then capture the particles. So you could eliminate one step of the purification scheme. But in the case of measles virus, this also could have been a challenge, and some of the other viruses, because we're still going through those steps of purification. And so Sarah Butcher's lab, at about the same time as us, was also looking at measles virus structure and a number of the other paramyxoviruses. And one of the things that both of us found, although it was in a smaller percentage of the particles that we were imaging, but in a larger percentage of the ones she was imaging, I can't address why that was, but if you look at the RNP here, it should just be a single herringbone lattice. So if you're looking at it from side view, it's a helical structure. And so what we both saw was that it was actually coated with another component on the surface. And so you can see that here and the structure here. We also did resolve areas where there was an uncoated RNP, so just a single herringbone lattice. And so what Sarah did with some really elegant immuno-EM and other work is to illustrate that this was the matrix protein that was coating the RNP. And this was a great model to think about, that the matrix protein could coat the RNP, but it also left us wondering, is this a real part of the virus assembly or maturation process? Or is this in some aspect maybe a destructive process that happens during purification of these virus particles? So if we think about paramyxoviruses in general, the more common theme of thinking about how they are structured is that you have the glycoproteins on the surface, the lipid envelope, the matrix protein lining that envelope, and the RNP within it. And so the model that Sarah proposed was that we have the glycoproteins, the lipid envelope, maybe there's some little snippets of matrix protein, but a majority of it is coating the RNP as a protective layer that helps facilitate its then transfer into the host cell when the virus fuses. But since we were looking in both of our cases at these purified virus particles, this gave us some pause to think, is this really what measles virus is doing to support its replication cycle, or is this an artifact that we have introduced? And so this is where, in the lab, basically most of the virus work that we were doing, either on HIV, measles virus, or respiratory syncytial virus, all nucleated around the process of doing this whole cell cryo-ET. So our first step in really going forward with any of these crazy experiments is to start off with understanding the cell types that we're studying. And so in the lab, because we're working on a lot of different virus systems, we basically test out any cell type possible that we could grow on an EM grid, determine if it can grow very thinly on the substrate, or if it's just going to not be useful for cryo-EM imaging. And so we do this by just doing this as conventional immunofluorescence microscopy, either on a MatTek dish or on a quantifoil grid, to validate that in XY plane you get good spreading, but in Z it's really critical, of course, to see what the thickness is. And there will be regions where we can actually capture cells that are thin, and that we can see these events of virus infection and assembly. This is also, of course, great for the correlative imaging pipelines that we're constantly pushing forward into the lab. So in kind, we also want to validate that as we are infecting these cells with virus, in this case this is RSV, but we have complementary experiments with measles virus, that we can culture the cells on a substrate. Now these are not grids, this is cell culture dishes or MatTek dishes, that we can culture these cells, that we can change the multiplicity of infections, so ratio between the number of cells to the number of infectious virus particles that are present there, and that we're not producing any, what we term defective interfering particles, so non-viable virus in the system. Because as you know with EM, if we were to go in at an MOI of 0.01, we would have very few events to image, and so we would be screening the whole world to find one virus that has infected the cell and is assembling. Whereas if we go in at a much higher MOI, not normal for people who are doing standard molecular virology work, this gives us more events to image in the microscope. But we have to play that game very carefully to make sure we're not introducing artifact. So this was exciting to see. We started off with HeLa cells, our typical model cell for any of these systems that we're looking at. And so the cell body, of course, which is way too thick for us to image through. But we have a lot of real estate on the edges and then these thin extensions that come off from the cells, where all along this region we can capture spots of virus assembly. So, one side of the equation that we're interested in. But we can also capture isolated virus particles that have already budded off from the host cell. So this is the perfect world for us because we have never touched those virus particles. So the structure of those virus particles is going to be their most native conformational state. So this is one of my favorite movies, or data sets that we collected. And so Josh collected this data. This is one of the first really spectacular measles virus assembly site data sets that we collected because it showed everything that we had been wanting to see in measles virus structure for the last 7 years at that point in time. And so we were finally able to, this is a snapshot from here in the upper right-hand corner, resolve the glycoprotein lattice. In this case, this was just F, the fusion glycoprotein, in this particular lattice arrangement on the surface. And then if we go under the lipid envelope, we were able to resolve the matrix protein lattice. And then finally, the RNP that was underneath the matrix protein. So we could really see this dynamic, positioning between the glycoproteins on the surface, the matrix protein there, and the RNP just below. Now, if we look inside view, it is really on micro scale here, so I apologize. But what was really exciting, all of it was, but if you remember the first data set that I showed you of the purified measles virus particle, where the glycoproteins were basically just this forest of trees, and you couldn't differentiate one from the other because they were just had been altered structurally. Now we can see every single H, so the attachment glycoprotein, these are H heads here, and then every single F head here, so the fusion glycoprotein. And so this gave us finally the place where we could come in and do subvolume averaging to resolve these structures. And so I don't think I have to really explain this here, but this was really the place where we could think about each of these structures distinctly, or maybe understanding how lattices superimpose on one another to really validate the structure of these particular targets in place. I can highlight subvolume averaging, so you cut out your volume. They're randomly oriented, you align them, you go through this process iteratively 1,000,000 times to then refine to get a higher signal to noise, higher resolution structure, ejecting particles, adding new particles. And so we did these for all 3 of these particular targets. And so the first one, in most of the work that we had been doing with measles virus or respiratory syncytial virus, we had not been able to resolve the matrix protein layer. And so this is really the anchor that tethers every single other complex in the virus as it's assembling or after it's released. I'm looking at a purified virus or released virus particle. And so we could see that it had this checkerboard lattice. Now, this checkerboard lattice had been seen before by Michael Rossman's group in Newcastle disease virus. And so we used, at the same time, they also generated the crystal structure of the Newcastle disease virus M and fit it into a lattice. Unfortunately, at this point in time, although there are people working on it, we don't have a crystal structure for a measles virus matrix protein. So this is why we use the NDV dimer to input it into our subvolume averages, and so Zhenlong put this in here. And we have some ideas of where there could be these interfaces that are holding the lattice together. So this gives us points where we could generate mutants to disrupt these lattices and understand how we alter virus assembly at this point, and potentially the incorporation of the other macromolecules within the virus. And so there's 2 dimensions to be aware of here. This 7.8 nanometer spacing in this direction, and then this 11 nanometer spacing in this direction of the lattice. So the next step, I know this doesn't seem like logical order, but the next step we want to look at is the glycoproteins on the surface. Now, if you recall, I mentioned that we have 2 glycoproteins, the fusion glycoprotein and the attachment glycoprotein, or H. So H is the taller one, but in actuality on the virus, if you're looking at wild-type virus, it's very hard to differentiate the height, so conveniently our collaborator, Richard Plumper, generated a virus that had a longer H stalk. And so he had done a lot of work. Measles virus is one of the major things he works on in the lab, to where we know that the F head interacts with the H stalk in this region of these particular amino acids. So he made a number of different viruses that extended the height of the stalk, either below this region or above this region. And those that he extended above this region were still infectious. Obviously, ones below it were not. And so it was this extended H virus that we did a lot of work on to understand the relationship space of the glycoproteins on the surface of the virus, because it gave us that height separation that we could resolve them independently of one another. And so if you remember that trimer lattice, what was really exciting for us to see, and this seems far too pixelated now, but you can probably appreciate it better here, is that in this cryo-ET data, we were resolving the individual trimers in their orientation space on the surface of this assembling particle, and that they were actually randomly oriented. So it wasn't like there were a bunch of triangles all like this. They were like this, rotating in different places. And this was related to some work that Kelly Lee had done, and this is negative stain tomography that he had performed on HPIV3, where he also resolved the trimeric structure of the fusion glycoprotein on those virus particles. So this gave us really nice confirmation that the work we were doing was pushing the limits. And so what Zalong did in this case was he did subvolume averaging of these lattices, either in these isolated patches or in patches where there was F and H present, remember at the different heights, to illustrate what the lattice and organization was of the F layer. And so what we found is that it also made this checkerboard lattice, and just conveniently it also had this 11 nanometer spacing that I highlighted before. Now, obviously, since we're looking at 2 different glycoproteins in these very complex systems, we wanted to validate that the structure we were looking at this particular layer was F. So Zalong went back in and did subvolume averaging of those isolated F glycoproteins to just validate that at that layer of that particular lattice, that we were only bringing F into the equation and not H at all. So we'll show this movie on the slide because I think that gives you a strong appreciation. So what you can see is the matrix protein lattice having this checkerboard lattice this way, but then we have F above it, and it also has this checkerboard lattice. So what's nice is we have the 11 nanometer spacing in that lattice, and we also have 11 nanometer spacing in the matrix protein lattice. So what's that's illustrating is that it's the staggered lattice appearance here. If we overlay them, you can see it in the movie there. What we are missing continuously in this lattice, and it caused us great pains in the lab, is we didn't see consistently regular spacing of the H lattice, so that attachment glycoprotein lattice, that's the upper lattice in the data. And so we have a couple of hypotheses of what's going on there. That it's possible that maybe H is not filling every single one of these positions, that it's a more altered distribution, or that if it is, it's possible that the F or H heads are so flexible, so there's heads up, heads down, heads up, heads down conformation, heads up, heads down conformation, and they can also be very flexible in space, kind of like a tree. And so it's just possible that with the subvolume averaging, we were not able to pull them out sufficiently. So if you have any ideas of how we could improve this, much appreciated, because we tried, or Zhenlong tried just about everything in the world possible to get that H lattice average. So the final structure that we used subvolume averaging on to understand in a little bit more detail was the ribonucleotide protein complex. So there's obviously been a very high resolution structure done of N of measles virus that gave us a model to then look at and cross-check that everything we were doing was okay. But in the context of the intact virus particle, what we found was the spacing in the helical pitch was the 7.8 nanometers. But if we were looking at the virus particles, we also saw that there was a lot of flexibility, as you can see here, in the RNP complex. And so Zhenlong went back through and did classification, and he was at minimum able to pull out two separate class averages from the full subvolume averaging, where the spacing in the helical pitch changed from 7.3 nanometers to 8.3 nanometers. And so a couple ideas we have as to why this is because these virus particles are very flexible, and the matrix protein is slightly flexing as well, is this is allowing the RNP to flex and move, and that helical pitch is changing based on what's going on with the matrix protein lattice. The other one could be that maybe at some point in time there is a compaction of this lattice or the helical pitch that takes place based on some aspect of the virus assembly process. What was cool, of course, is after we do these classes of the subvolume average, Zhenlong could then fit them in to see even along different stretches of the lattice that you can incorporate different points of that helical structure. So we really need to look at a lot more particles, I would say, so that then we can enhance the number of classes and see how dynamic the structure is overall. But what I want to illustrate here, which is the second last slide on this one, is that when we look at this coordination of the RNP to the matrix protein lattice, you can see that they're very related to one another. So if the RNP is running there, the matrix protein lattice is directly above it and is oriented in the direction, or they're oriented in the direction of one another. It's not a haphazard arrangement. And so Zhenlong, what we're showing here is that this is coordinated, so you can have lattices going in multiple different directions of the matrix protein, and that's regulating the position of the RNP within the virus particle. And there, of course, is an orientation based on the polarity of the RNP with respect to the matrix protein. So a next step in this process would be to go in, get a higher signal-to-noise resolution structure of the RNP to then be able, and the matrix protein lattice, so then we could understand what are the connection points between the RNP and M that are regulating it. Because you remember, or maybe not, I didn't highlight it again, is the spacing of the matrix protein lattice in one dimension was 7.8 nanometers, which that helical pitch of the average was also 7.8 nanometers. So we know there is a direct linkage coordination space with respect to them. So in summary, we still use the affinity capture technique for a lot of different samples in the lab. We work on other retroviruses and things, and so this is a very useful tool for eliminating some of this debris that's present. May not be the best approach we're using for RSV and measles, because we have now demonstrated that this purification seems to really disrupt particle structure. With using this whole cell cryo-ET approach, we were able to finally resolve the matrix protein lattice, and it forms these arrays which then coordinate the glycoproteins above the membrane and the RNP complex below the membrane. As I mentioned, H is definitely present there, but we haven't been able to resolve how we're doing the subvolume averaging to help us pull out that structure in a bit more detail. So, all of this work was done while we were still at Emory, and it was a really tour de force amount of work from Zhenlong, Josh, Rebecca, Sherry, and Gabby. Or Gabriella was the first one to really start working on measles virus in the lab, and she did a lot of that affinity capture work. But it was really these four team members that really pushed that forward and helped us generate this paper and this work. We had great collaborators with Richard and Melinda, who gave us every virus we could ever dream of and purified them at constant levels until we figured out that that wasn't the best path. Of course, I think everyone appreciates the work of David Mastriani because we collected all this data with serial EM. We were using Joule microscopes and direct electron detectors for all this work, and of course the alumni. We've moved, and so we haven't started measles work yet, but we're getting the RSV stuff going. So I'm happy to take any questions. Thank you. Uh-oh. No, you're not allowed to ask questions.
So the distribution of the matrix protein, whether it's associated with FNH or with the RNP, could that be part of the maturation of the virus, and perhaps there's, as it matures, it associates more with the RNP, and kind of like the way the HIV matures, or is that not just,
So now I don't know if I should put all these slides in. Thing. So the matrix protein, when the virus is assembling, coats the entire membrane and then released virus particles. But one of the things we look at is the virus at different points in time. So 48 hours after infection, 72, whatever. And so that's a whole distribution of particles on the grid at that point in time. And so if we're looking at later stages of virus infection,
maybe you already said it.
No, I didn't, because this is one of the things that was still a curiosity to us. So if we look at very late virus particles, we do see the matrix protein coating the RNP, which is what you're asking. But in those cases, so that means the matrix protein has left the membrane. And so in those cases, what happens is that the fusion glycoprotein has then triggered. And so it's not in the pre-fusion conformational state, it's in post. And so Long went through both for RSV and measles virus, because this was a concern point for us, to validate were those particles still infectious or not. And they had a lower infectivity level, either in RSV or measles, when the F glycoprotein had triggered. So if we look at them here, I know they just look like little dots, but there's much more dense population of pre-fusion F versus post, and so we calculated that. So he also did some other experiments, which are kind of more, I don't know, they weren't as beautiful as we would have liked them to be, but it shows where the infectivity goes down with heat treatment, so we can force this triggering event to happen at F, which then releases M from the inside. And so in this case, in the particles, we don't see any M on the membrane. Well, we see very limited M on the membrane, but most of it has actually come away and is forming these aggregates. And so we don't know if that's an in-between state, and then it'll coat the RNP or not. So this requires a lot more work to resolve this. But we do feel like it's a non-functional path. Since
there's some allosteric mechanism between F and N that is probably important for infection, and you're seeing in these particles what looks like a particle that's just kind of associated with the cell, basically, ready to infect.
Right.
Exactly.
So we know the cytoplasmic tail of F interacts with M. RSV and measles, and so that's where we could look at some mutants in this realm to see if we disrupt this, and then we get maybe a triggered population versus not, and then how that relationship space happens. So yeah, stay tuned. That's an ongoing project.
Thank you for your nice talk and very beautiful work. I would have a question about the RNP because it seems to be a very flexible structure. And have you seen any differences in the areas that are bendy and the straight parts, or is it like in structure-wise? Is there more spacing, or
Yeah, sure. So
is it more condensed in the straight regions?
So if we look at the ones where they're more flexible, like here, you know, the edges, there will be, I mean, just thinking about how something is,
Yeah.
they will be flexed out more.
Mm-hmm.
And so when we were focusing on our subvolume averaging, we were going for the straighter segments. So we would have to think about how to straighten those more flexible segments if we wanted to incorporate them into averages. So there definitely is that, and we can see that with how the matrix protein is on above the membrane, or above the RNP.
It's not that there are different proteins associated more to the straight areas than to the curved areas.
You mean matrix or just other things
Yeah.
in general? Right, the stretches of matrix where there's a straight RNP are more regular than when it's starting to bend.
Thank you.
Yeah, I have a question related to this. So, is the genome in this case, is it linear inside this RNP helical arrangement, or is
No,
it?
it
Do you know
wraps.
that?
It wraps around.
Can you actually more or less resolve it in subtomographies? Or can you,
Not at the resolution of these subvolume averages, but maybe in
but it wraps
future space.
like in the inside of this, like what is it, helix?
So it's coordinated within, and
Mhm.
it's wrapping within. So we can't see it in
Yeah.
in these densities. Now we have in some, not in measles virus, but on RSV, there's some cases where at sites of assembly, it appears like we're seeing the RNP complex assembling, and we actually see little RNAs,
Okay.
the genome there,
Mhm.
the filament.
Yeah.
But not in this case. It's too dense.
Yes. And how big is one RNP entity? Do you know this?
One RNP. I have totally spaced out.
Yeah. No. I can do that.
What's the sequence of the Measles virus formation then? Is it that first for example, the matrix protein marks the site where one would be then rising, or is it all just like jumbling together and just blood off at some point?
So the order of recruitment.
Exactly.
So we don't have a good answer to that question yet. There are a couple of theories. I mean, clearly, if you're thinking about, well, glycoproteins have to get to the surface, that they should be there first, and then the matrix protein comes in afterwards. So typically, if we see the glycoproteins, I mean, it's just us getting the time course analysis, and so most of the time, our events we're looking at have been later, so we're not capturing those initial states. Now that's some of the work we're starting to work on with labeling and different techniques, so that we can answer this particular question. I think they're kind of, well, it depends on the virologist you talk to. I think many of these things are kind of co-recruited, and so it's forming a complex at the same time, because typically in all of our data, there's a site of assembly, everything's there, and we're not just seeing pieces haphazardly in the cytoplasm.
So you then assume that the matrix protein kind of like is also part of the recruitment of the RNPs to the nascent lab as well.
Thanks.